In this lecture, we'll be discussing cell membranes. This material is found in chapter 11 of the book. The title slide really kind of shows a depiction of a membrane and all the various pieces that you find in the membrane. And as we'll learn today, there's a lot more than just the phospholipids that make up the membrane. Specifically, what we'll be talking about, first we'll go through some composition of membranes, detailing what phospholipids are in them, sterols, and also proteins. Okay, all three of those make up membranes. Okay, it's not simply just lipids. Then we'll talk about the dynamics of membranes, and they're actually very fluid structures, and we'll introduce what's known as the fluid mosaic model. And then finally, we'll talk about transport. In transport, there are a few different types of transport we'll cover. Basically, how membranes allow certain things to go through them and how they keep certain things inside or on the outside. First, we'll go through some review questions from chapter 10 material. Okay, and the first review question, rank the following fatty acids from the lowest to highest melting points. So think back to chapter 10 material, remember what affects the melting points. Right? It might be helpful to pause the lecture and work through this in a couple minutes. Okay, so we have 20, 0, 21, delta 12, and 23, delta 12, 15, 18. So let me select my pen. Okay, and if you remember, how are melting points affected? Well, they're affected by chain length and they're affected by the number of double bonds. And in this case, all three of these have the same chain length. They're all 20 carbons long. Okay, so that's not going to be affecting these. Okay, and I'm just going to call these, we're going to call that 1, this 2, and this fatty acid 3. Okay, for simplicity. So which one of these has the lowest melting point? Okay, and if you remember, low melting point means they're more fluid. They're going to be at a given temperature, it's going to be more in the liquid phase. Okay, so the number of double bonds, as that increases, the melting point decreases. Okay, so 3 is actually going to have the lowest melting point. Okay, because it has 3 double bonds. 2 only has 1 double bond, so it will be next. And then finally, 1 will have the highest melting point because it has no double bonds. It's saturated. Okay, second review question. Draw the mixed triglyceride with 12, 0, 14, 1, delta 10, and 16, 3, delta 7, 10, 13. Okay, so this question, probably not going to see this on an exam, although I guess I could ask you to draw this and submit it electronically. But if we had to do that, let's think back to how a triglyceride works. Well, we have first the backbone of this is a glycerol. And if you remember, there's actually 3 carbons in a glycerol. And I might as well draw these hydrogens in explicitly, although you don't really need to. Okay. Sorry, when I put my hand down, it makes marks on the screen. Okay, so glycerol would normally have 3 alcohol groups on it. Right, so those would be OHs normally for glycerol. But in a triacyl glycerol, or triglyceride, those are going to be connected to the carboxylic acid via an ester linkage. Okay, so we can draw our first fatty acid here. Okay, this we'll put 12-0 at this point. So that's carbon 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, and that one doesn't have any double bonds, so it's pretty easy to draw. Okay, next we'll do 14-1 delta 10. Okay, we draw our ester linkage. And now we draw our chain, 14 carbons long, a double bond at position 10. Okay, so that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We make our double bond, 11, 12, 13, 14. Okay. And then finally we have 16, 3, delta 7, 10, 13. Okay, carbon 1, 2, 3, 4, 5, 6, 7. Double bond, 8, 9, 10, 11, 12, 13, 14, 15, 16. Okay. So that's basically how you would draw a triglycerol, triacyl glycerol, or triglyceride. Right, so if you were, a possible online exam question, right? If you're doing a paper exam, you could, expect to have a question to draw one of these, I guess. But for an online exam, I could ask you, give you a structure of one of these, and then have you name all three of the fatty acids in it, I suppose. Okay, and then last review question, name these lipids. Right, so this might be a point where you'd want to pause and see if you can name all of these. Okay, if we go through this, okay, what is this? Well, it has a choline as the head group, but you'll notice also there's a very short acetyl ester here at position 2, and there's an ether linkage here. So if you remember what this one is, it's platelets activating factor. Okay, let's look at B. B has a phosphocholine as a head group, okay, and an amide linkage here. Okay, and we don't have this isn't really a fatty acid here, where we have an alcohol sticking out. So if you remember, this is actually sphingosine, this part of the molecule right here, with the amide linkage and the phospho head group. That's sphingomyelin. Okay. C, this is cholesterol, or I believe the slide this came off of was actually 7-deoxycholesterol, something like that. The one that is a precursor to vitamin D, but really, if you see this structure, you can, in this chain here, you could just think of this as cholesterol. D, phosphocholine head group, and these are ester linkages. So this is what we would call phosphatidylcholine. E, this is a cartoon picture, and I wouldn't expect you to know the exact name of this. This is A antigen in the blood group. But what it is this, if you remember this kind of cartoon depiction here, we have one fatty acid. This is sphingosine. So this is and sugars here, so it's a glycosphingolipid. All right, F, this lipid here, highlighting the cis double bond configuration between 11 and 12. This is 11 cis retinal. Okay. involved in site. Okay, and then the head groups, if we go through, if we can name these, right, we have 2 carbons and a amine group, so that's ethanolamine. 2 carbons, then the nitrogen with methyl groups around it, and again that should have a plus charge. The book figure does not show that, but it does have a plus charge. That's choline. Here we have serine, glycerol, and phosphatidyl inositol. So that this would be the inositol head group, okay, which I wouldn't really expect you to know this one. Right. Okay. Moving on to chapter 11. So what are membranes? Well, they're complex lipid-based structures that form pliable sheets. Okay. They're composed of a variety of lipids and proteins. Some membrane lipids and proteins are glycosylated, meaning they have sugars attached to them. Okay, all cells have a cell membrane. That's what makes them a cell. It separates the cell, the interior of that cell, from its surroundings. Okay. Eukaryotic cells have other membranes inside them. They have internal membranes that sort of divide into organelles that are inside the cell. Okay. All membranes have 3 important properties. Okay. 1, they are flexible. Okay, they have to be flexible because they need to permit shape changes, right? Cells have to be able to grow, or they have to be able to move, so they need to be flexible. If your membrane was rigid, they would just break apart and spill the contents, right? So they need to be flexible, right? The second important property, membranes are self-sealing. Okay, that means 2 membranes can come together to form one, to fuse together, and that's a process known as exocytosis. Membranes can also undergo fission, that is, they can divide into 2, right? And so cell division, that's very important. The fact that membranes can be divided into 2 separate compartments. Right? And finally, the third important property, they are selectively permeable. They retain certain compounds inside, and they exclude other compounds from the outside. Right? That's really important, because if they weren't, if there wasn't some selectivity in them, the contents inside would be the same as the contents outside, and that really wouldn't be conducive to life. This picture shows how many membranes are inside a eukaryotic cell. We have the plasma membrane would be sort of on the outside of this, but this is showing all the membranes inside. You have the endoplasmic reticulum, nuclear membrane, mitochondria have their own membranes, and then there's other compartments within the cell that contain membranes, like these secretory granules. Okay, so there's a lot of membranes going on inside a eukaryotic cell. Okay. Functions of membranes. Well, first and foremost, it defines the boundary of the cell. Okay, the outside plasma membrane gives the cell its shape and its boundaries. Okay. They also allow the import and export, okay, and that goes back to that property of being selectively permeable. Okay. They can selectively import things like nutrients, right, you need to get into the cell, and you can selectively export things like waste products from metabolic pathways, and also toxins. Okay. Another function of membranes is that they retain metabolites and ions within the cell. Okay, that still goes back to being selectively permeable. Okay. Membranes can sense external signals, and they can transmit that information into the cell. And we'll talk more about this in chapter 12, signaling pathways. But things like like an external signal, external hormone or growth factor can reach the outside of a cell membrane, and that signal can be conduced to inside the cell nucleus, and a specific gene then can be expressed. Okay. And membranes, help facilitate this process. It's sort of where that process starts for the cell. Okay. Another function, they provide compartmentalization within the cell. Okay. And you can think of this as being that kind of self-sealing property. And that's important because you can separate energy producing reactions from energy consuming ones. Right? If you have these reactions right by each other, it'll be kind of like a futile cycle. You're producing energy and using it all at once, and that's not always good. So like the ATP producing reactions inside a mitochondria, right? Those are within the mitochondria, okay, and they're separated from reactions where you would be using that ATP energy. Okay. You can also keep proteolytic enzymes, so enzymes that digest proteins, away from the important proteins that need to be that you want to protect from being degraded at a particular time. Okay. Some common features of membranes: they're sheet-like, flexible structures, approximately 30 to 100 angstroms thick, so that's a pretty big range of thicknesses. Okay. The main structure is composed of two leaflets of lipids, and we call this a bilayer. We'll see pictures of this in a second. Okay, so membranes are usually, for the most part, you can just assume that all membranes are actual bilayers. We did mention the archaebacteria in chapter 10 when we looked at lipids from them. They're actually a monolayer, but their monolayers mimic the bilayer that are seen in all the other membranes. Membranes form spontaneously in aqueous solution. Okay? So there's no enzyme or machinery needed to build the membrane. It forms spontaneously, and that's primarily due to the hydrophobic effect, which we've discussed. Okay. Protein molecules are present in the membranes, in all membranes, and some protein molecules can span the entire lipid bilayer. Right. Membranes are asymmetric, and that what we're meaning there is that some lipids are found mostly on the inside of the bilayer, on the inside of the cell. Some lipids are found on the outside of a bilayer, or the outside of the cell. Carbohydrate are always on the outside of a cell. So if the glycosylated lipids or proteins are going to be on the outside of the cell. Okay. Membranes are also asymmetric in that they can be electrically polarized, and typically the inside would be the negative side. Right. They are fluid structures, okay? And you can think of them as kind of a two-dimensional solution of oriented lipids. And that's where this fluid mosaic model comes in. Right? It was proposed in the early 70s. Okay? And what it states is that lipids form a viscous two-dimensional solvent into which proteins are inserted and integrated. You can also think of it just in simple terms. The fluid mosaic model is proteins are in a sea, floating in a sea of lipids. Okay, that's really a simple way to state the fluid mosaic model. Okay. Integral proteins are firmly associated with the membrane. Oftentimes they span the entire bilayer. Peripheral proteins are more weakly associated and can be removed relatively easy. right? Some are non-covalently attached, and others are covalently attached to lipid membranes. Excuse me, membrane lipids. Okay. So a very cartoon sort of depiction from the book of the fluid mosaic model, right? We have the polar head groups of our membrane lipids. These would be phospholipids, are shown here in blue. Okay, and the hydrophobic tails are shown in yellow. Okay, so this would be one bilayer, and this one faces the outside of the cell. And another bilayer is shown here, and the head groups face the inside of the cell. Okay, so this is what we mean by bilayer, right? The hydrophobic tails face each other on the two layers of this. So you have the outside and the inside surface are polar, and the middle of the bilayer is nonpolar. And then we have various membrane proteins, which we'll talk about in some more detail, but here you have one that's an integral membrane protein. It has a helix that spans the entire length of the bilayer. And then you have an example of a peripheral protein that's just associated with the polar head groups of the membrane. Okay. So the composition of membranes. Well, as we saw in that picture there, there's going to be a component that's lipid based and a component that's protein based. Okay. The composition of membranes is going to be different. It's different in different organisms. It's different in different tissues from the same organism, and it's also different in the different organelles within a single cell. Okay. What changes? Well, the ratio of lipid to protein can vary. The type of phospholipids can vary. Abundance and types of certain sterols varies. For instance, prokaryotes, bacteria, really don't have any sterol in their membranes. Okay. Cholesterol is very prominent in plasma membranes in our cells, but it's really almost absent in our mitochondria. Okay. Galactolipids, so lipids that have sugar groups attached, are abundant in plant chloroplasts, but they're really almost absent in animals. Okay. This table here, you don't have to know any of these values, but it just shows you the components by weight percent of various membranes. Okay, so myelin sheath here, we have 30% protein, 30% phospholipid, and almost 20% sterol, in that sterol being cholesterol. Right. Then you can see there, they do change. In E. coli, for instance, it's almost 75% weight protein in its membrane and only 25% weight phospholipid, right? So the amount of protein in a membrane is actually very, very high. Here's another figure showing this more graphically, how the composition of membranes can change. Okay, and these are looking at different organelles. In looks like rat liver cell membranes. Okay, so here's the plasma membrane. In orange is cholesterol, so it has a huge component of cholesterol in that membrane. Right, and blue is phosphatidylcholine. The darker blue is phosphatidylethanolamine. Some minor lipids, and then green is sphingolipids. Okay. So you can see that there's the plasma membrane has a lot of sterols. Mitochondrial membranes hardly have any. Okay, and they have this red component, which is cardiolipin. Okay, so really the point here to make is that different organelles are going to have fairly drastically different membrane compositions. Right. Another way membranes are asymmetric is that, or that they can differ, is they can be asymmetric from the inside of the inside monolayer and the outside monolayer. And this figure shows that. So this part of the plot is the inner monolayer. This part of the plot is the outer monolayer, and you can see that they're actually quite different in composition. Right. The outer leaflet typically has more of a positive charge, okay, and that's reflected here with phosphatidylcholine and sphingomyelin. Both of those have an overall positive charge to them, okay, and they make up the majority of the outer monolayer. Phosphatidylethanolamine and phosphatidylserine are the overall negatively charged amino acids, and you can see that they are in a higher amount on the inside monolayer. Okay, and that's not by accident. Okay, that has some biological significance to it. And when you see phosphatidylserine going to the outside monolayer, that has some signaling implications. Okay, so one, depending what kind of a membrane you're in, if it's in a platelet, phosphatidylserine on the outside will activate blood clotting. Other cells, when phosphatidylserine makes it to the outside in a large amount, that marks the cell for destruction, for apoptosis. Right, and it's kind of another figure showing this. So if we look here, the outside monolayer, we're going to have more sphingolipids, more positively charged lipids, and more cholesterol, or sterol. The inside is going to be predominantly phosphatidylethanolamine and phosphatidylserine. Okay, so that's on the plasma membrane, the outside membrane of the cell. The inside membranes, these vesicles, they're kind of going to be reversed. Okay, if you'll notice, the outer membrane of the vesicle looks like the inner membrane of the plasma membrane, right? And that's so they can associate with each other when they connect. Okay, and on the inside, that's where we're going to get the positively charged head groups in the cholesterol. Okay. Right, and in a similar with the Golgi body. Okay. So all membranes have some asymmetry to them. Okay. If we look a little bit closer at membrane proteins now, okay, and this figure kind of gives us an overview of what we have. We're gonna see throughout this lecture some examples of proteins that span the entire membrane, and some that have channels through them that can actively pump things from one side to the other, or just allow the diffusion of things through them. Then there are some that are buried in one side of the membrane or span the entire membrane. In various ways, we'll see different examples of those. And then we have things that are peripheral proteins that are only associated with one face of the membrane and not actually buried inside of it. Okay. So some functions of proteins that are in membranes. Well, you can have receptors, okay, and those detect signals from outside the cell. Okay, some various receptors are shown here, some examples. A light receptor known as opsin. You can have hormone receptors like the insulin receptor. Neurotransmitter. Acetylcholine receptor is an example of a neurotransmitter. Pheromones, things like taste and smell receptors would be examples. Okay. You can also have channels, gates, and pumps. Okay, so nutrients, ions, the potassium ion channel and sodium ion channels, and then also things like neurotransmitter, serotonin reuptake protein acts to pump serotonin back into the cell. Okay. And finally, enzymes. We don't really see many examples of these, but some enzymes can be membrane proteins. And in metabolism, you'll talk about ATP synthesis, and that the enzyme for ATP synthesis would be an example of an enzyme being associated with a membrane, being a membrane protein. Okay. This is the figure from your book showing membrane proteins. Okay, we're going to look at these in a little bit more detail. Okay, peripheral membrane proteins. Okay, they associate with polar head groups of the membranes or charged regions of integral membrane proteins. So in this example, we have a positive charge region of this integral membrane protein, and this peripheral protein is associating with that. Okay. They're relatively loosely associated with the membrane through ionic interactions with either the lipids or the domains of other proteins. And because they're sort of loosely associated, they can be removed by fairly gentle conditions if you disrupt the ionic interactions, either by adding higher salt concentrations or changing the pH. You can get these peripheral proteins to be released. So if you're trying to purify a peripheral membrane protein, things like adding salt or changing the pH, you can purify. You can remove those peripheral proteins and then purify them. Okay. When you do purify a peripheral membrane protein, they will no longer be associated with any lipids. Okay. The other type would be an integral membrane protein, and those are ones that are buried in the membrane, in the hydrophobic region of the membrane. Okay. They can span the entire membrane, so this example here would be a protein that spans the entire membrane, and it has a portion that sticks out on each side. Okay. They have asymmetry just like the membranes do, so different domains will face different compartments. Okay, so there's a directionality towards these integral membrane proteins. Okay, they're very tightly associated with the membrane, and that's because they have hydrophobic stretches in the protein that interacts with the hydrophobic regions of inside that membrane bilayer. So in this case, this integral membrane protein, this yellow part of it is the hydrophobic region, and it associates very strongly with the hydrophobic region of the bilayer. Okay. To remove an integral membrane protein, that requires the addition of detergents. Okay. And detergents are molecules that have a very hydrophobic part and a hydrophilic part. So if you add enough detergent, what that's going to do, it's going to act to pull a protein out of a membrane, because the hydrophobic parts of the detergent can stick to the hydrophobic part of the protein, and then that hydrophilic part of the detergent can be water soluble. So it really acts to pull that out and solubilize that protein. Okay, so the detergents are similar to the phospholipids, but they're maybe a little bit shorter, smaller, okay, easier to solubilize. Okay. A purified integral membrane protein is still going to have phospholipids or detergents associated with them, okay, to be soluble. So when you purify an integral membrane protein, it'll have this sort of detergent micelle around it. Okay, so it's not ever, to get rid of that. In a sense, if you got rid of that, the protein is going to fold differently to have that hydrophobic region not exposed to the polar solvent. Okay, and it'll fold differently and likely precipitate out of solution. Okay, an example of an integral membrane protein that we've talked about already in this class is bacteriorhodopsin. Okay, and this one, this is the 3D crystal structure of bacteriorhodopsin shown in a kind of a cartoon membrane. Okay, there's the C terminal will stick to the inside of the cell, and the N terminal will be on the outside of the cell. So there's a directionality to it. Okay, and really don't need to know too much about this protein specifically, other than it is an integral membrane protein, and those 7 helices basically span the entire length of the membrane. Right, there are 6 types of integral membrane proteins, depending on the directionality they have or how many times they span the membrane. Okay, type 1 is a protein that spans the length of the membrane and has the C terminal inside the cell. Okay, and the N terminal outside. 2 is the opposite, type 2. The N terminal will be inside, the C terminal will be outside. Type 3 integral membrane proteins are those that have multi regions that span the membrane. So in this case, we have 1, 2, 3, 4, 5 helical regions, and they're all in the same polypeptide chain. Okay, so that's type 3. Okay, jumping to type 5, very similar to type 3, we have multiple membrane spanning regions, but they're all on different polypeptide chains, right? And these polypeptide chains just associate together to form a complex that the integral membrane protein needs to function. Okay, so these are multimeric type 5. Okay, type 4 are those that have a phospholipid anchor. Okay, so they're anchored into the membrane by a phospholipid, and the protein part is almost exclusively either on the inside of the cell. Probably most of the time, type 4 here are the protein's going to be facing the inside of the cell. Okay, but they're considered integral because they have a hydrophobic anchor. Okay, as opposed to being a peripheral protein, they actually have a piece of them buried in the membrane, so they're harder to remove. You can't just add high salt concentrations to remove that. Okay, and then finally type 6, I don't know if you'll see any examples of these, but these have a protein that spans the membrane and then another region that's anchored into the membrane via a hydrophobic interaction. Okay, so amino acids in membrane proteins are themselves sort of asymmetric. They cluster in different distinct regions. Okay, and as you'd expect, charged residues tend to cluster on the aqueous exposed, so either the inside or the outside portions of those integral membrane proteins. So charged residues are shown in blue, and you can see for the most part they're facing either inside the cell or outside the cell. Hydrophobic amino acids obviously would be in this region where the hydrophobic tails are inside the bilayer. What might not be as obvious though, tyrosine and tryptophan tend to cluster at the nonpolar and polar interface. So tryptophan here is in red, tyrosine in yellow, and you can see that they're very much focused, almost in some of these proteins, almost like a line, right where the interface in the membrane between the polar head groups and the hydrophobic tails is. Okay, you can think of those as forming kind of a seal, like a grease seal or something, right? They're big aromatic side chains, primarily hydrophobic, although tyrosine does have an OH group on it. But those big sort of ring structures are good to make a sort of a seal, if you like think about it that way, from the aqueous portion to the hydrophobic portion. Okay. Now let's talk about some physical properties of membranes. So we talked about the composition. Now let's look into the physical properties. Okay, membranes are very dynamic structures and very flexible structures. And remember, that's one of the important characteristics of membranes, is that they are flexible. Okay? They can exist in various phases and undergo phase changes, actually. Okay? They're not permeable to large polar solutes and ions. Okay? And that comes back to them being selective. So big polar molecules and ions can't readily pass through a membrane. They are, however, permeable to some small polar molecules and nonpolar compounds. Okay? And that again goes back to them being sort of selective. Okay? Permeability can be artificially increased by some chemical treatment, and this is a way that we can get DNA into certain cells. Okay? We can add DNA with certain chemicals that makes it easier to pass through membranes, and that's in the lab. That's how we do some bioengineering to study the effects of certain genes inside cells. Okay. So let's talk about the phases of membranes. Okay, depending on the composition and the temperature, lipid bilayer can be in what we think of as two different phases: a gel phase or a fluid phase. The gel phase is more like a solid. Okay, individual molecules don't move around as much. In the fluid phase, the individual molecules can move around, fairly freely. And heating causes the phase transition from the gel to the fluid phase. Okay, under physiological conditions, membranes are more fluid-like than gel-like. Okay, so they're more liquid-like than solid. And that's important because they have to have some fluidity to them for proper function. Okay. And just saying, gel or solid, and a, liquid or fluid phase, either or is a little bit simplistic. As we'll see, there really are kind of a mixture between the two phases. But the book gives these two pictures of this. So, and they call it a liquid ordered state, okay, and a liquid disordered state. So the liquid ordered state you could think of as being like more like the gel state or a solid state, and a liquid disordered state would be like a fluid state, true fluid state. Okay, and there's, this, as temperature increases, you go from a more solid-like or liquid-ordered to a more disordered or fluid-like state. Okay, and there's some transition here. Okay, and it turns out that under physiological conditions, membranes are really kind of floating along in this transition region. So they're not completely solid or gel-like, and they're not really completely fluid or disordered. Okay, they're kind of a mixture of these two phases. Okay. What influences this fluidity of membranes? It's mainly by the fatty acid composition. Okay. More fluid membranes require shorter and more unsaturated fatty acids. Okay, and remember the difference. We sort of talked about that at the first review question in this lecture, right? Increasing chain lengths of the fatty acids is going to lower, raise the melting point. Okay, so at higher temperatures, cells need to have more saturated fatty acids or longer chain fatty acids. Okay, and that's to maintain integrity. you can't have, if you get too fluid, your membrane's going to be, too leaky, and it won't have the integrity needed. Okay, so at higher temperatures, you're going to have more saturated fatty acids in those membranes, or longer chains. Okay, and at lower temperatures, the opposite is true. You're going to need to have more unsaturated fatty acids or shorter chains, and that's to maintain some fluidity. Because if the membrane gets too solid or gel-like, things can't move around like they need to. And this table, obviously, you don't have to memorize these, but it shows you, it's pretty interesting. This is the composition of E. coli cells grown at different temperatures. And here are a list of the different fatty acids found in the membranes. Okay. And you'll see that these two are unsaturated fatty acids. These two are saturated. Okay, so as we raise the temperature, the percentage of our saturated fatty acids increases, right? That's to maintain integrity. Okay, as the temperature decreases, you can see that the unsaturated fatty acids, the percentage there increases, and that's to maintain fluidity. Right. Another very important molecule in terms of membrane dynamics are sterols. And in the case of eukaryotic cells or animal cells like we have, cholesterol is that sterol. Okay. And sterols, like cholesterol, help maintain rigidity and decrease permeability. So they're sort of twofold. Right. And other organisms like plants and fungi have their own versions of cholesterol, different types of sterols. Right. So how does cholesterol work? Well, it's very planar, and it has very hydrophobic. That steroid nucleus there is very hydrophobic, so they kind of position themselves almost like plates between the fatty acid chains of the phospholipids. So they can kind of stack in between there. Okay. Cholesterol does two things, and they're sort of kind of opposites of each other. Okay. In one sense, it helps prevent the crystalline or gel or liquid ordered. All of those mean the same thing. It helps prevent that state. And you can think of that kind of as lowering the melting point. Okay. It also increases membrane packing through this interaction with the phospholipid fatty acid chains. Okay, and that reduces membrane fluidity, and that helps also to prevent leaks. So in a sense, that raises the melting point. So it both lowers the melting point and raises the melting point, which is weird. Okay, I'll explain that in a second here. The other thing it does is it's important in the formation of what are known as lipid rafts, which we'll talk about. There's certain regions in the membrane that are sort of more ordered, and they have certain types of phospholipids, certain proteins in them, and they float around almost like rafts. Okay, so how can it both raise and lower the melting point? Well, in a sense, it doesn't. But what it does is it would flatten this region out here in the curve. Okay, so instead of being this huge, very abrupt transition, the curve might look more like this, where you have a flatter line through this transition region, which allows the membrane to be more, in the correct amount of fluid-like versus solid-like in a physiological temperature range, right? Where here, if the temperature changes all of a sudden, your membrane's going to drastically change in phase. If this line was flatter, that cholesterol really makes happen. That gives it a bigger temperature range where it has the optimum fluidity. Okay. Membrane dynamics. Let's talk about how the membrane becomes asymmetric and how it stays asymmetric, right? We can watch individual lipid molecules as they move what we would call laterally within a single bilayer. So if we look at this one, it can move relative either in two dimensions in a single bilayer. Okay, and this would be uncatalyzed lateral diffusion. That happens very fast, something like 1 micrometer per second within a leaflet. Okay. Okay, every component of the membrane exhibits some sort of asymmetry. Okay, lipids, there's outer and inner leaflets have different lipid composition. Proteins, as we mentioned, certain peripheral proteins are only associated with one side of the membrane. Integral membrane proteins have different domains on different sides, so they insert in the membrane in a certain direction. Lipid modification of proteins targets the protein to a specific leaflet. Okay, so proteins are asymmetric. Carbohydrates, again, as we mentioned, they're only on the outside of the cell, so they're also asymmetric. Okay. Now let's look at if we talked about a membrane, let's say this is the outside of the cell, flipping over to the inside of the cell. Okay, so flips from a leaflet to another are rare, right? So uncatalyzed, so we'd call this flip-flop diffusion. They're very rare because you have that polar head group has to go through the hydrophobic region of the membrane, and that's obviously pretty unfavorable. So the half-life of these a lipid doing this is something in the order of days. So that's a very slow process. We have proteins called flipases in membranes that help facilitate this process. Okay, they catalyze transverse diffusion or the flip-flopping, if you want to think of it that way. Okay, some of them use ATP to move lipids against their concentration gradient. So an example of this flipase, right? It's using ATP to move a lipid, and in this case, it's the phosphatidylethylamine and phosphatidylserine from the outer leaflet to the inner leaflet. Remember that phosphatidylethylamine and phosphatidylserine are more concentrated on the inside leaflet, right? So that if you had your membrane form spontaneously, the amount of different phospholipids on the outside and inside, they're going to be about the same. But you have these enzymes, these flipases, that can move the phosphatidylethylamine and phosphatidylserine to primarily the inside. Okay. So that's a flipase, moving something from the outside to the inside. A flopase would be the other direction. You're moving something from the inside to the outside. Okay. And in this case, it moves. It would move like phosphatidylcholine from the inside to the outside. So they're more prominent on the outside, and this is why. Okay, and then there's other things called scramblases that will move one lipid from the inside to the outside while it moves another lipid from the outside to the inside. Okay, and so that scramblase probably don't really see any examples of that specifically. Okay. All right, so the flipase and flopase is really how the membranes can get their asymmetry and maintain their asymmetry. There are two important experiments that show membrane dynamics that we'll talk about, and they're covered in the book. Okay, one is known as FRAP, or fluorescence recovery after photobleaching, and the other is just simply a single molecule labeling experiment. Okay, so we'll go through each of these individually. Okay, so the first FRAP, okay, this is the overview. Okay. So first we take a cell in this experiment and we label the entire outside with a fluorescent probe that attaches to the head groups of these lipids. Okay, so the whole entire outside of this cell, it would be, if you viewed it under a fluorescent microscope, it would all light up. Okay, next you hit this outside of a cell with an intense laser beam. Okay, so bam, you hit it with the laser. What that laser does, it's intense enough that it kills those fluorescent groups that were attached to the membrane where that laser hits. Okay, so you get what known as a bleached area. Okay, so it removes all of that fluorescent color, that fluorescent molecule from that area where the laser hit. Okay. Then you wait a certain amount of time and you watch how the fluorescent grows back into that area. Okay, and the only way that fluorescence can grow back in that area is that a lipid molecule that has an intact fluorescent molecule, an active one, can diffuse back into that region. Okay, and by doing that, that's how they measured the rate of diffusion of these lipids within a bilayer, and they could see that it was fluid. The fluorescence did come back. It didn't just stay bleached. It actually came back. Okay, and the only way that could occur again is if those lipids are fluid and moving. Okay, the second experiment, a single lipid was labeled fluorescently, and then they watched its motion. Okay, and it would kind of bounce around, move around, and it would stay sort of, within a certain time frame. It would stay in a relatively enclosed region, and then all of a sudden it would make a jump, and then it would bounce around in sort of a different region. Okay, and then it makes another jump, and it stays in this localized region for a while, and then there's another jump, and then it's in another region. And I went backwards here, so this is where it started and this is where it finished, but that doesn't really make a difference here in the results, right? The results showed that there were these regions where the lipid could be more mobile, and it have to wait a certain amount of time. It took longer to get to another region. Okay, so it wasn't just kind of moving around randomly all over the place. There was these defined regions it spent time in, and they kind of thought of this as sort of like there being a fence around each of these regions that the lipid had to take a while to jump over. Okay. All right, and that's where this idea of membrane rafts came from. Okay. Lipid rafts contain clusters of certain phospholipids, and in particular some glycosphingolipids that have longer than usual tails. Okay, what those do is they're more ordered. Okay, and those lipid rafts you can think of as being more of a gel or solid phase because of that. Okay, so those would be more of a solid region. Okay, and they have specific proteins that interact with those regions, that kind of you can think of sitting on that raft. So these more solid regions that we call rafts kind of float around in the more liquid part of the membrane on the outside of them. Okay, and they carry certain proteins that need to be in close proximity to one another, and they do contain slightly more cholesterol in those regions as well. Okay. And so those rafts are what you can think of as the fences in this experiment, right? That this lipid moving around in the fluid part is probably bouncing into some rafts or other integral membrane proteins around this that kind of restrict its motion. And then every once in a while it'll get past one of those and move to another region. Membrane fusion, membranes can fuse together, and that's without losing continuity, which is very important. If a membrane had to fuse and it let things leak out when it was doing that, or leak in from the inside, that would obviously be very bad. Okay. This fusion can be spontaneous, or it can be mediated, controlled by proteins. Okay. And some examples of protein-mediated fusion are entry of viruses like the influenza virus into a host cell, or the release of neurotransmitters at nerve synapses. Right, and these, you won't need to know examples of these, but if you're interested, there's, exocytosis is when something, a vesicle from the inside of the cell fuses with the plasma membrane and lets that out. Endocytosis is the opposite, where you have something from the outside of the cell kind of create a bubble in the plasma membrane, which then turns into a sort of vesicle on the inside. Okay, viral infection, right? We've mentioned that before. That virus connects with the membrane somehow via a receptor or something that the virus is targeting that's on the outside of the cell, and then it fuses its membrane with the cell membrane and injects, its DNA or RNA into the cell. Okay, sperm cells and egg cells react by fusion, okay, and then there's other sort of internal fusion mechanisms as well. Neurotransmitter release, that's another example of the protein-mediated fusion, and those are by the snap and snare proteins. And again, this is outside of the scope of our class, but if you're interested, there's, at least at one time fairly recently, there was a lot of research going on with these snap and snare proteins. Okay. Moving on to the third section, transport across membranes. Okay, cell membranes are permeable to small nonpolar molecules. Those can passively diffuse through the membrane. Passive diffusion of polar molecules is more difficult because it involves first the desolvation, the getting rid of the water molecules that are associated with a polar molecule. Right, so they have a very high activation barrier to get a polar membrane through a polar molecule through a membrane. Okay. Transport across membranes can be facilitated by proteins that provide alternative paths, diffusion paths. Okay, such proteins are called transporters or permeases. There's as we'll see here on the slide, 6 different types that we'll sort of talk about of transport. Okay, one is simple diffusion. Okay, simple diffusion. In this case, we're looking at something from the outside of the cell diffusing in. Okay, this only happens simple diffusion for non-polar compounds. Okay, and it has to move down a concentration gradient. So that the concentration of S has to be greater, in this example, on the outside of the cell than the inside for it to passively diffuse through that membrane. Okay. Facilitated diffusion is something that moves down an electrochemical gradient. Okay, but it can be a polar molecule now, but it's being helped across that membrane by a channel, a protein that will, help it, and we'll see an example of this in a second. It just helps lower the activation energy of that polar molecule moving across the nonpolar membrane. Okay. So those are diffusion. Okay. The next examples that we'll see are active transport. So one is primary active transport, And that's where you're moving against an electrochemical gradient. So in this example, S is going to be a higher concentration on the inside of the cell, but you're still moving it from the outside to the inside. Okay? And you do that using ATP as that driving force. Okay, so you're using ATP to actively, the hydrolysis of ATP to actively push more of this S solute molecule inside, or substrate molecule, I should say, inside. Okay. Secondary active transport, again, you're moving S against an electrochemical gradient, but that's driven by some other molecule or ion moving down its electrochemical gradient. So in this example, this ion will be in much higher concentration outside of the cell. So it will want to move, there'll be a electromotive force to move it to the inside of the cell. And you're coupling that energy of this, doing this spontaneously, this spontaneous movement of that ion with the non-spontaneous movement of your substrate going from a low concentration into a higher concentration. Okay. Ion channels. Okay, these are passive things that an ion can move through that channel down its electrochemical gradient. Sometimes they're gated either by most commonly by a ligand. So they'll be closed until a certain ligand binds to them, and then they open up to allow passage. Okay, and then something that we really won't ever see an example of in the class, but ionophore mediated ion transport is when an ion would bind with another more hydrophobic molecule, be brought into the cell, and then that ion would be released. But I've never seen an example of that in class. Okay. Right. So this is if we're looking at a polar molecule just simply diffusing through a membrane, what that would look like. Well, it has a sphere of hydration around that polar molecule. Okay. So the polar water molecules are interacting with that polar molecule. For that polar molecule to diffuse through the membrane, all those water molecules that are associating with it will just have to, break off and separate out. And that actually, if you're looking at the free energy of that to break those interactions of the water molecules, you pay a pretty high free energy price for that. Okay? And as you're going through the membrane there with your polar molecule, that's also a pretty high free energy because your polar molecule can't interact with any of the hydrophobic groups. Okay. Then on the other side, again, you're going to need to recreate that hydration sphere. So the free energy of that process is shown here in red, and that delta G of the transition state is really high. What a transporter does is when that shell of hydration breaks, there'll be amino acids here in this channel that are polar that will interact with that polar molecule. And what that does is it just lowers that free energy required to lose the water molecules, and now you're not interacting. You don't have this polar molecule going through a hydrophobic region. You have some other polar interactions it can have, and that drives down this transition state energy. And remember, when you lower transition state energy, what you're doing is you're speeding up the process, right? That in kinetics, that's how enzymes work. So you're speeding up the transport. Something that would could take years or longer for a polar molecule to break through that membrane can now be done in seconds or milliseconds. Okay. So there are three classes of these transport systems, and they're based on the directionality to them. One, you can have something called a uniport, and that's when you're moving one molecule one direction. Okay. You can also have co-transport, a couple different types of cotransport, and that's when you move two molecules at once. Okay, so a symport is when you're moving two molecules in the same direction, and an antiport is when you have two molecules moving in opposite directions. Right. Some specific examples of transporters that the book talks about that we'll highlight here. One is aquaporins. Okay, what aquaporins do is they allow water passage through membranes, and although that's not really obvious, they are really important to do that because if you have, in the case of like a bacteria cell, if its environment that it's in becomes more salty on the outside or less salty than the inside of its cell, that would put a tremendous amount of osmotic pressure on that cell, and if it doesn't have a way to normalize the osmotic pressure between the inside and the outside of the cell, that cell could burst, right, and then the bacteria would die. So one way that cells get around this is by aquaporins, and these allow very rapid movement of water molecules through membranes. Okay, and they do this in a number of ways. Okay, and the book goes through some specifics here. Okay, you have these helices that are charged, that have a charge gradient on them. Okay, and they actually have some, negative charges on the outside and a positive charge towards the inside. What that does is it gives electrostatic repulsion. So certain an ion isn't going to be able to go through there because it's going to be repelled. Okay, this is referred to as the ARR selectivity filter. So the small AR is aromatic residue, and the big R is the arginine positive charge residues. Okay, so those make the selectivity filter, so only water can pass through. Okay, they also contain two NAP motifs, and those are used to move these water molecules single file through here. Okay, and that this is just the abbreviation of the amino acids and the motifs, so asparagine, alanine, proline motifs. And we see the asparagines in the two motifs here. Okay, and water molecules move through this very orderly and in single file. They move through these channels, and you can see that each step they have some hydrogen bonding interactions. And what else is interesting is that they go in with the oxygen, facing inside on the channel that they're going through. The oxygen molecule will face, in this case, it would be facing down, and then at a certain point here, it flips and reorients, and then the oxygen molecule would be facing up as it comes out this side. Okay, so there's some very specific things that this channel does with water, and when it does that, it can move these water molecules through very, very fast. Okay, and I included this Wikipedia page. Peter Agri was the one that did a lot of research on these aquaporins, and this website aquaporins.org is something, if you're interested, you can look and find a lot of information about these. Right. Another example of a transporter that we've mentioned before in this class was this in the bicarbonate cycle. Okay, in blood cells you have the carbon dioxide. It's relatively nonpolar. It can diffuse through the membrane. Okay, once it's in there, an enzyme called carbonic anhydrase converts it into bicarbonate. And then you have this transporter in the membrane that can move bicarbonate to the outside. It actually works in both directions, but in this example on this side, it's moving bicarbonate to the outside, and it does that at the same time it moves a chloride ion from the outside to the inside. So this would be an antiporter. Okay, it's moving, it's doing co-transport to things at the same time, but they're moving in different directions. Right, if we look specifically now at active transport, okay, remember there are two types. We have primary active transport, and that's where we're moving one thing in one direction, and it's using ATP to do that. Okay, so we're moving, in this case, S1 is lower concentration inside the cell, higher outside, but we're moving it to we're continuing to move it to the outside against its gradient by hydrolyzing ATP as our energy source. A secondary active transport is when you use the movement of one thing with its concentration gradient. So here S now is moving with its concentration gradient, and we're doing that along at the same time we're moving something else, S2, against its concentration gradient. Okay, so this would be an example of a symport because it's moving two things in the same direction. Okay. Oftentimes, this secondary active transport, how is it generating the concentration gradient of S1 that's needed to move S2? Well, there's another channel for S1 that's using ATP. Okay, so you're consuming ATP to pump S1 outside of the cell, right, against its gradient, so you can use it coming back in this symport to import S2. Okay, so that would be secondary active transport. Okay, ion channels. We'll be talking more about ion channels. They maintain gradients for active transport. Okay. An example here is the potassium ion channel, and what you'll notice is they have on the outside they have helices that are there's a charge gradient on them. Okay, and their negative side is facing more towards the inside here. Okay, and that negative charge is going to help stabilize the positively charged potassium ion that's going through that channel. Okay, so that's one way they're specific. The other way in which they're specific is the channel size here is very specific for the size of the potassium ion. And if you look down the outside of it, down the middle of it, you'll see that, right, that channel there is really anything bigger than that potassium ion isn't going to fit. Okay, and other things that are smaller than it aren't going to get the same interactions. And they're specifically the interactions come from the backbone carbonyl oxygens that are facing in this channel, and they act to as a way to sort of hydrate these potassium ions, right? Because when they go into that channel, they have to lose the water molecules that are coordinating with them, and those interactions are replaced by the interaction with the backbone carbonyl oxygens when they're inside the channel. And the spacing of those is such that they only really interact with potassium precisely. Okay. Right, and there's other ion channels, right? There's sodium and calcium ion channels as well that act in kind of a similar fashion. And this table is showing you the certain channels there are. There are many of these channels, and when you have one of these genes that are affected by a mutation, there are certain diseases that are involved. And probably the most famous one, ion channel defect, would be the chloride ion channel, which results in cystic fibrosis. Okay, so next time we'll move on to chapter 12, and we'll cover really kind of the first part of chapter 12, sections 1 and 2. Chapter 12 is dealing with biosignaling. We'll talk about the general features and then talk about G protein coupled receptors. And then we'll jump to section 10, which it covers a specific G protein coupled receptor, and that is involved in vision. Okay, and a reminder, our third in-class activity will be October 22nd, so make sure you show up for that class meeting.